Thank you very much. Vinod?
Going back to the last slide, you said you had to make a comparison between the test and the reference product.
That's right.
Sorry, the summary slide.
Okay.
But what we are really looking, if there is no reference product, this is a compounded product which is not existing before. So how do you do the comparison in those cases? How do you test the efficacy and the safety of the product?
So that's right. I agree that not all the compounded products would have an exact reference product. But per se, if you have a combination of diclofenac and lidocaine and something else, probably a compounded product can be compared against an approved diclofenac gel and an approved lidocaine cream or whatever has been approved as an innovative product because that has already gone through the clinical trials and proven to be effective.
No, but some of the cases it is not gone through. This is the brand new ingredient in the compounded topical pain. So how do you do the comparison?
Yeah, so I don't know. Like that's something that we need to set up a standards on.
Okay.
I have no clue.
So in other words, at least still for, as I see it from your presentation, you need to do the in vitro testing, and that is the drug release.
Exactly.
And the permeation testing for the drug penetration to make sure that the compound will be effective as a analgesic or whatever it may be. Is that true?
That's correct. It's not like you need to do both the tests on all the products because even in case of FDA guidelines, if you see if it's a simple solution, semi-solid solution, FDA would only ask for IVRT testing. And if it's a complex product like acyclovir cream and such things, so there is a requirement of IVPT. And some drugs you would also may have to do a PK study besides IVRT, IVPT, right? So it depends on the level of complexity associated in the product. You would determine what kind of tests need to be performed.
Our complexity is the cream. We are not looking at solutions. We are looking at the cream preparation, compounded pain cream. So it's a complex product.
That's correct.
So what testings would you suggest we need to do to at least have some kind of an assurance that it will be effective?
Well, again, the same thing. It is like if you have a reference product, you definitely can compare your compounded product against multiple approved reference products in terms of release and permeation. These are the two main tests that we generally perform to find out if our generic product matches with the reference product.
I think John had his card.
Yeah. So very complicated process with multiple factors. Reminds me of trying to cook souffle or something else where it depends on how you mix it together and what temperature you put it at and all the rest of that. And so for that, there is a few factors that go into it. It's called a recipe, and I assume there are also recipes that go into the creation of various solutions and creams and other things, as well as some of the equipment that you showed, having specific equipment to try and standardize this in some way. And I guess what I would want to know is that from you or from your colleagues or in a journal article or others, is there a careful definition of how such products should be produced that might be then usable by a compounding pharmacist, or that might be helpful in understanding whether what's being created will at least be standardized, if not more or less effective, depending on testing?
Yeah, good question. Again, formulation is a complex science. You have to have a thorough understanding of the excipients, their physicochemical properties, and how they behave when they all come together. So you only learn by trial and error process. That's why, you know, product development in industry takes like several years, and testing is another phase. I don't know if there is any shortcut way of arriving at a lead product in the compounding.
I guess what I'm asking is once you or colleagues or other scientists have figured out what the right way is to make a particular product or a particular, now obviously it could go in a formal route through FDA approval and become diclofenac gel or something like that. But if there is an understanding about which excipient to use for which types of drug APIs and what temperature to be done at and how much mixing, is some of that published somewhere so that a pharmacist would be able to find that and then use that to create a product that is more consistent, at least?
There's plethora of literature available on the excipients and the type of formulations that they that they come together and form, and the impact of such formulation microstructure on the permeation of different kinds of drugs. So there's a whole lot of literature, but I think the compounding pharmacists are not really exposed to the side of science, because I teach in the school. I know there is only one semester we teach pharmaceutics, which is all formulation. So we kind of give them the ready recipes and make them do just to know the science of formulation, not really in-depth knowledge on all those excipients and process and such things.
And so just as a last question, this may actually relate to this previous speaker, but is there a website somewhere that sort of has a list of the appropriate formulations of some of these things? Is there a place a resource, a textbook, something which you can go and look things up?
Yeah, there are quite a few books on the formulation science. There are several websites, and there are some formularies from different countries available for the pharmacists to refer to.
All right. So you're describing, how you can do this, well, sophisticated, in vitro, permeation testing, right? But, I'm wondering in regard to all these compounded products out there. So you describe how you do it, but who actually does it? Who actually does that testing? I mean, in this compounding process, does a compounder do it? Do they go to you and ask you to test it for them? Or, I mean, when does this testing actually come into play for these products that we are talking about?
Right, so that need to be figured out for sure. Now that Sarah was talking about this outsourcing facilities which comply with all the CGMP guidelines and stuff like that, which is an FDA approved facility, possibly that is a place where one can set up all this evaluation, experimental setup and do all this type of tests, I guess. Right,
but essentially right now there's no requirement for it, right? I mean, there's not like all those products that are out there being marketed have all undergone this kind of testing.
That's true, yeah,
okay.
That's right.
All right, and you did show this table, with all the active ingredients and the different columns in regard to, I would certainly love to hear more about that and get that all explained a little bit further. But the question is really where John came in. Where can we look this all up, right? How do we even, if we want to assess this, how do compounders who actually are the pharmacists who produce this, where do they get the information? And now we heard about the training that is being offered. But given the vast, the great variety, the heterogeneous nature of all these products, and then knowing that it makes such a difference in what kind of solvent, what the non-active ingredients are in order to, and they need to match basically the active medication, I'm kind of having trouble. We have 3 to 6 medications in there.
No.
And we have one solvent typically, or maybe several one way, but how can we get a solvent that is optimized for each of these ingredients when they're all so different? And that leads me to the question, are there really good combinations? You know, active ingredients that we know have the same kind of solvent and would work better together. And again, where do we look that up? And do compounders who actually make these products truly take all that into consideration as those make those decisions?
Yeah, not that I'm aware of that the compounding pharmacists would have knowledge of all this aspects of interaction between the drugs and drug and excipients and stuff like that.
So I just wanted to follow up quickly on the same point. That was a really excellent point. I was going to ask something similar. Is there at least literature on which drugs would be best applied with which of the excipients for dermal penetration? Is that knowledge even assembled somewhere?
There's a lot of literature reports on per se lidocaine. People developed lidocaine creams with PEGs and with poloxamers, with other kind of fatty alcohols and stuff like that. So definitely there's a lot of literature. Mm-hmm. Whether it's a head-to-head comparison of all these things, that's rare to find.
Mm-hmm. Or is anybody like organized, like here are all the different types of solvents you might use or things you could put in a cream and which drugs are best solubilized or would penetrate best with those types of ingredients? Is that assembled anywhere in any review articles?
Not really. Not like an educating thing to a pharmacist.
Uh-huh.
Just from the basic science perspective, somebody's interested in making a cream out of fatty alcohols, and they would study, like in this case, I just studied different fatty alcohols and its influence on the permeation. But I never bothered to compare a clotrimazole hydrogel, prepared with a polymer against a cream that is prepared with fatty alcohol. That was not my interest.
So, okay, is there enough knowledge in the basic literature to assemble that information?
There's quite a bit of information available in the literature that a person who has expertise in the formulation science can put together and probably, educate the pharmacists, compounding pharmacists.
Then I guess
that's a possibility.
Yeah, one would then have to test when you combine them all together.
Yeah.
What happens then? So I won't take up any more time. Can you all separate out who was first? I didn't pay attention because I was... Okay, I'm just going to go in the middle. Carmen.
So, interesting talk. Thank you. You talked about your in vitro permeation testing, and I wonder, you talk about human skin, and there's a lot of variability in human skin. Whether or not you chose, it looks like the standard is cadaver skin.
That's right.
And so I would ask a couple of questions, and I'll just field them out there for you, throw them out there for you. Is there a difference between cadaver skin and living skin? Is there a difference between abdominoplasty skin that's you may have gotten recently out of the operating room bin in comparison to the skin that may be on someone's arm or on their chest wall? And so how does that relate if you're going to give something, something like lidocaine, which may have the abilities to prevent or even cause arrhythmias? Then I would ask the question as follow-up as to whether or not a person's skin color, how much exposure they have to the sun, their age, so the pediatric, a neonate versus an elderly person, or the amount of hair they have on their skin might play, as well as things like their gender. So, we know that women may change as it relates to, where they are in their menstrual cycle and things of that nature. So how much of that information is known? And then I would ask about what that means in the context of someone who might have injured skin, such as someone who is burned or someone who may have CRPS that we're trying to apply this cream to. And do these things, so can you answer that question? And then, so why don't you answer that question? I think that's big enough. And then I have a follow-up in regards to it. I think I know what the answer might be, so it may be quick.
Sure. Well, in real life situation, if the skin has a cut or injury, then there is no stratum corneum. Mainly, stratum corneum is the main barrier for the drug to penetrate into. And if the stratum corneum is gone, then it's an open skin, basically, right? So when we do in vitro permeation testing, we take care of all these things. We do a quality control on the skin. The skin that we use are tested for what is called as transepidermal water evaporation rate. Okay, they have to be of certain standard, and we also check the electrical resistivity of the skin. So we choose the skin which is intact and not injured or does not have any signs of other kind of diseases and such things.
So how does that relate to the next question? So you chose intact skin, but some of our patients don't have, that we might be prescribing this for, don't necessarily have intact skin. Are there sort of warnings? Well, you don't put these things together, but do you put those as limitations moving forward?
Well, so the main objective of this test is just to evaluate the topical formulation in terms of its ability to deliver drug into the skin and maybe across the skin, right? So we have, particularly in case of FDA-approved products, we are evaluating a generic product which is compositionally similar with the reference product. So all we need to do is we need to select intact skin, pieces of skin, and subject both the products for evaluation on the same donor skins, run the studies simultaneously, and compare the data. If both of them perform comparably, then the generic product is going to match, in clinic as well, in terms of its clinical efficacy is the general assumption.
That's the assumption.
That's right.
Okay, so when does that assumption fail?
When would that assumption fail? See, I'm not sure if I understood your question. If you ask me, like, when would that assumption fail? Because when the two products, whether it is innovator product or the generic product, goes on to a skin that is injured, that is hyperkeratinized or having some other kind of pathology, it would not perform the way it should because of the differences in the skin, huge difference in the skin permeability. So regardless whether it is a reference product or the generic product, there will be potential failure modes because of differences in the skin permeability.
You asked a question that I
Yes,
think
can you
he
follow,
didn't
please?
answer, which
Yeah.
Neonatal skin, children's skin, adult skin, older people's skin. Can you help us to understand, is there a gradient across them, or is it distinctly different? If a product is put on a young child versus put on an older person, would we expect it to be different?
Sure. From the literature, I have not tested personally, but from the literature, people have shown that skin from a neonatal skin generally would be highly permeable because the stratum corneum is not completely formed. And once the stratum corneum is formed, I think that takes place right after a month or 2 months after the childbirth. After that, the permeability would be similar to an adult skin. Okay.
And old people, as they get older, the skin gets much thinner.
Yeah.
Are the features of the skin the same in terms of permeability, or?
No. If the skin undergoes any kind of changes due to aging, the thinning, or maybe the intercellular spaces opening up more, then it would become more and more permeable. So the geriatric patients' skin is relatively more permeable. That's correct, as compared to the adults.
Okay, Deborah. Oh, sorry. We're running out of time.
You mentioned earlier something about PK and doing formal PK analysis. That brings me to my question. What is the role of blood levels in these kinds of analyses? In other words, to look at the whole intact organism and what gets across the skin, does it make it to the systemic circulation?
Could you come again, please? The question is not clear to me, please.
Your talk is about testing and in vitro testing.
That's right.
And at what point would you also do in vivo testing where you would look at actual blood levels? And what's your knowledge of how often that's been done with these kinds of preparations?
Well, on the formulations that are compounded, I'm not aware of any such studies being carried out. But otherwise, for the products that are submitted for approval to FDA, the products that contain APIs that would work by different mechanisms, that would have different target sites of action, are generally required to be subjected to pharmacokinetic evaluation. So that is kind of routinely done. Like, for example, Dapsone cream, that requires a PK study, IVRT, IVPT, and PK study. But an acyclovir cream would be just IVRT and IVPT.
So as a pharmacologist, I think I know that you're saying, therefore, you need formal PK blood level studies,
right.
sometimes tissue level studies. I just want to clarify that because not everyone here is a pharmacologist.
Mm-hmm.
Quick question and follow-up to the previous discussion.
Sorry. So the question I had is about skin. And is there a standard area of the body that is used?
That I use for
Skin.
my testing.
Because in my experience, skin thickness is not the same.
That's right.
But maybe that doesn't have anything to do with stratum corneum.
That's right, yeah. Generally, I use the back skin or the thigh skin.
But that's a standard in the whole nation. I mean, everybody in the world uses a particular part.
Well, you have to use the skin from the same region when you're comparing two products. That is the requirement as per the guidance, FDA guidance.
Okay.
I generally prefer to use the thigh skin or a back skin because I get sufficient area of skin so that I can,
So that's
evaluate
important.
both the products or maybe more products. If there are more generics.
Okay, Carol.
In the in vitro testing, could you give us an idea of just generally how well the Franz diffusion cell works to give a somewhat comparison? Obviously, for pharmacokinetic study, blood levels are going to show you on an intact person best, depending on, of course, variability, inter-individual variability. Then probably cadaver skin would be next, and then pig skin, and then a full in vitro, for like a Franz diffusion cell. Could you give us a quick insight on how those compare? So if you're measuring something in a Franz cell, how close is that going to be to blood level?
Well,
I just don't know how well that system
No,
works. I know it's used a lot. I just don't know how well it really works.
I know the question is about in vitro and
Besides
vivo correlation.
qualitative versus quantitative.
Right. So, well, to a certain extent, the skin permeability studies would be able to correlate with the in vivo. Most of the drugs would. But in case of in vitro release testing, I don't think there is any such possibility of correlation with the PK data. Right.
Okay, it's my turn anyway. So many years ago, I chaired a Pharmaceutical Science Advisory Committee meeting at the FDA, and Vinod was at the FDA, and it was joined between our group and the dermatology group. And they rejected almost anything except a direct clinical trial. They said that the cadaver skin is not accurate, skin stripping is not accurate. And from what I recall, and we may need to get some dermatologists, and maybe Vinod can come in on this, but what I recall is they talked about follicles and permeability and variability of permeability due to hair follicles. Do you want to comment on that? How big a role would hair follicle differences in different people's skin play a role?
Sure. FDA, I think, was approving the skin tape stripping technique earlier to determine the amount of drug present in the stratum corneum, and later I think that particular guidance was withdrawn. I don't know the present status on the tape stripping technique. But in vitro permeation testing has been there since quite some time as one of the requirements. Probably Dr. Shah would be able to comment on this point.
Okay. Coming back to Steve's question, and also Carmen asked you, the skin varies from individual to individual. Even in the same individual, in different parts of the body, the skin permeation characteristics are different. And therefore, for the evaluation purposes, you are taking the skin from the same area, one area, and making the comparison of the two products, as he referred to as test and the reference, in the same system at the same time. So all the other variabilities are excluded because you are comparing the same type of skin at the same time. Now, I agree what Steve indicated, that when I was there, the skin permeation methodology for the bioequivalence was not accepted. It was rejected primarily because of lots of variability and the problem with measuring the drug permeation characteristics. But I think for the purpose of looking into the pain medications, coming back to our situation, making a comparison or having some kind of an indication that drug does penetrate through the skin is an important factor. Because for any topical drug product to be effective, there are three principles it should take place. First, the drug must be released from the formulation. Otherwise, you can't get it. That's why he showed initially the drug release experiments. Then it has to penetrate through the stratum corneum, the top layer of the skin, in order for it to have the functions inside, do the pharmacological activity, so that if the drug penetration or the permeation study is shown, and then if the action. So those are the things I think you evaluate the skin permeation only just to see whether there is going to be any drug going inside the stratum corneum and epidermis and dermis. Otherwise, if it does not go, there is no analgesic activity that would be affected by this particular type of the dosage form. And it will vary from formulation to formulation. As he asked, there are several different factors that will influence the drug penetration. More drug may go in depending upon the different formulation. As he showed, two formulations, the same ingredient but formulated differently, it may function differently. It may not be the same. So those are the important factors I think that we need to take into consideration. Steve? Yeah, sorry.
Before we go on, we're now out of time. Should we continue on with this line, or should we move to the next session?
So looking at our schedule, we've just been chatting briefly.
Yeah.
I think we can go, we took a very short break, but I think we may have a lot of questions for our next group.
Okay.
And so if we chose to take a couple more questions here, finish up,
Okay.
and bring our next panel up straight away. If we finish early, great. If we don't, we'll have plenty of time to ask the questions we need of the people that are actually doing some of this work.
Okay, so maybe another 5 minutes, and then we'll, yeah, at most. Okay, John?
A very short question, I hope. What's the order of magnitude difference between different parts, between different populations and different parts of the body with regards to absorption? I understand that there's a great deal of variability, but could it be 100-fold different? Or could it be, if I put it on my temple and put it on the bottom of my foot, obviously there's callus on the bottom of the foot. Could there be, if you can give me just a sense as to what range we're talking about, whether it's 2-fold, 5-fold, 10-fold, 100-fold, and then across different populations, African American or Caucasian or Native American, et cetera.
Right. So from my reading the literature, what I have seen is it varies from molecule to molecule. The variability is anywhere between an order of magnitude to 2 orders of magnitude.
So I just had a quick question to follow up on Steve's. My understanding of follicles is that that is not the bloodstream, and that things may go from the stratum corneum into the follicles, but they may not necessarily be absorbed. So how much does having more follicles versus less follicles matter for drug absorption?
Again, there's a lot of literature that shows that follicular pathway does not really significantly contribute to drug delivery into the skin. So it should not really matter a lot. It's a matter of the surface area of the stratum corneum available, what we call as active diffusion area, that really matters.
Yeah, and I think where it came into play was in tape stripping, because you might be pulling part of the follicle, I guess, or the entrance to follicles. I don't know. And so that would throw that method off for determining penetration. Any other questions before we finish this?
We heard a lot now about, how, what kind of changes can happen depending on what the non-active ingredients are. But I'm just wondering if there are some medications that will never make it into, through the stratum corneum into the dermis, epidermis. Basically, if a molecule is so large, or if it is so, ionic, maybe some, even with the best, vehicle and solvent, will never make it in there. So I'm just wondering if of all those medications that we've heard, are there some that you could highlight that will just not make it in there anyway? Or the amount of medication that gets, even with the best kind of, compounding, would still not probably go in there.
Yeah. Well, the formulation science has evolved so much. So there are ways and means to work around the permeability issues. As long as the molecular weight stays less than 600 Dalton, even if it is not fulfilling all the criteria, the API characteristics, we would be able to get it into the skin. The question now is like how much would be effective. Whether we will be able to deliver effective amounts of drug is the question, right? So some molecules may be poorly permeable, but they may be active at nanomolar level. So you may be able to formulate a successful topical product. Some drugs may be highly permeable, but they are required in milligram quantities in the skin to elicit pharmacological activity. You would never be able to get a clinically efficacious formulation, right? So all these things need to be put together.
Yeah, at this point, let's take a 5-minute break. There was one question from the audience. Do we want to take that?
It's a very quick question, which is, just as there is PDUFA funding by sponsors to evaluate dossiers, I wonder if people had any thought about the use of an elective or non-mandatory payment by a sponsor to delineate extra care that was taken about the source and purity of the APIs or excipients.
Sorry, what was the question again?
You could support, through the FDA, the process of tracing the individual components of a mixture. So it wouldn't be a complete black box, but there would be some certification of it. Maybe not through FDA, but I don't know. But in other words, you would think that a sponsor would have a monetary interest in being declared that it was certified by some agency or some impartial group, but then they would support the cost like some type of a seal of approval, if you will. I'm just throwing that out as a possible way to try to address the near total absence of data for non-FDA approved topical pain creams.
I guess
Yeah,
that's
it's
question,
difficult.
that's a question for any of the individuals who have spoken today, perhaps.
Yeah, it's difficult with no requirement to do so. So we'll now take a 5-minute break and be back at the top of the hour, or maybe 3-minute break.